Shotgun sequencing is a type of a sequencing technique in which it's kind of a methodology. So what we do here is we break up the DNA into short fragments and then we sequence them. It's called shotgun sequencing because it has an analogy with the random firing patterns of a shotgun. Here is general view. Shotgun sequencing is divided into two types. So one of them is clone by clone shotgun sequencing, and the other one is whole genome shotgun sequencing. In clone by clone, which is also known as hierarchical shotgun sequencing, we actually do that. We use some specific mapping techniques also. So the target DNA is first analyzed by clone-based physical maps, and then obviously the clones which overlap with one another, then they are selected and then they are subjected to shotgun sequencing. Here is a brief view of this clone by clone shotgun sequencing. So we take the genomic DNA and we take its roughly the sizes, we break them into the sizes roughly like around 200 megabase pairs. So these fragments are then broken down into smaller fragments and then they are cloned into some cloning vector. So normally we use YACs and BACs for larger genome sequencing. They were used for in the Human Genome Project also. Then we take these clone and we analyze for the presence of some landmarks which later on help them in order to find their relationships among one another. And the overlapping clones are called as contigs. So what we do next is we take these clones, the clones which are kind of minimally overlapping. So we don't want the clones that are kind of completely overlapping from one another in order to avoid redundancy. So we take minimally overlapping clones and then we purify those BACs and YACs for the DNA and then we physically fragment them. So in the end we get small fragments from these clones, and then these clones are again subclones into vectors. So we get those fragments from here and then we put them into the vectors again. So it's kind of again we have a second step of cloning. Then we get those clones and then we break them and then we present them to the sequencers, and then in the end we get those short reads. So actually the sequenced fragments coming out from sequencers, they are called as reads. So we take these reads and then these reads are assembled. There are different assembly packages or softwares. We'll talk about them later. So after that these reads are assembled together and then these assemblies are finished and then finally we have the final assembly. In whole genome shotgun sequencing, we take these DNA fragments and clone them into vectors and then we sequence them. So here we take the genomic DNA, we break them into the fragments of 500 to say 1000 base pairs and then we simply subclone them and then we take those subclones and then we get those sequencing reads just like we did earlier. Reads are then assembled into contigs and then contigs are then arranged into scaffolds and scaffolds are analyzed for the landmarks. Basically these are different markers that can guide us whether our assemblies are correct or not. And then in the end these scaffolds, they join together and they can give us the genome map. There is another technique which is called as hybrid shotgun sequencing. So that is kind of a mixture between these two. So we have those back shotgun reads and we can also have the whole genome shotgun reads, and we put them together and we look for the reads which are overlapping between these two and we retain those leads. So in this way we can have a stronger evidence and we can have the reads which are common to both of them and we use them for assembly, and then in the end we can expect a good finished product. So we see that in shotgun sequencing the DNA is broken down into the smaller fragments, cloned into the vectors, and then these segments are sequenced, and after we get those sequencing reads we put them together in a process called as assembly, and then in this way we can assemble the final genomes.